Hello everyone, welcome back. So we have been discussing on the initial phases of Krebs cycle, and we saw that the acetyl coenzyme A, which is a very important substrate in that enter the Krebs cycle, it is being produced from various substrates, including fatty acids, different kinds of amino acids like ketogenic amino acids, and the most important one among them, it is a pyruvic acid. The pyruvic acid, it is produced as a product of glucose oxidation, and it is one of the most important substrate for the production of acetyl coenzyme. This pyruvate, which is being produced as a result of glycolysis, the glycolysis it takes place in the cytoplasm. So pyruvic acid is produced in the cytoplasm, and this pyruvic acid it is transported into the mitochondrial matrix, uh, and then it is there where the pyruvate is getting converted to acetyl coenzyme. Okay, so the conversion of pyruvate to acetyl coenzyme takes place inside the mitochondria. Right. Now, the conversion of the pyruvate into acetyl coenzyme, it is an oxidative decarboxylation process, that is oxidation as well as decarboxylation process, and it consists of three steps. The three steps it is being catalyzed by the enzyme pyruvate dehydrogenase complex. Commonly it is referred as PDH. The pyruvate dehydrogenase complex, it catalyzes three steps which occurs in sequence. The first one, decarboxylation, the second, oxidation, and third, the transfer of the resultant acetyl group onto coenzyme A to produce the acetyl coenzyme A. Okay, so pyruvate undergoes decarboxylation, the intermediary substrate it undergoes oxidation, and then it undergoes a transfer, it undergoes what you call the transfer of acetyl group onto the coenzyme. So this is how the whole process takes place. Okay, we'll look into the detail of each of these steps, and this is how the final what you call the equation which shows the conversion. That is pyruvate, it gets converted to acetyl coenzyme A, right? And for that, along with pyruvate, coenzyme A is required, and NAD is getting utilized. NAD is getting utilized as a what you call oxidizing agent. Okay, then the coenzyme A pyruvate, it will react to produce acetyl coenzyme A and carbon dioxide, and NAD it will get reduced to produce NADH and H plus. Okay, so the pyruvate dehydrogenase complex, it contains three enzymes, that is pyruvate dehydrogenase, it is considered to be E1 in the equation over here, dihydrolipoyl transacetylase, that is E2 over here, and dihydrolipoyl dehydrogenase, that is the E3. Okay, so as a first step, the pyruvic acid it undergoes decarboxylation. It gets decarboxylated to produce, here you can see, it is the acetyl TPP complex. Okay, it undergoes decarboxylation, and the enzyme involved is pyruvate dehydrogenase. The product produced it is acetyl TPP complex, that is, it is an activated acetaldehyde, and carbon dioxide is released. So you can see here, so it is that is why it is known as a decarboxylation process. Okay, it is pyruvate dehydrogenase. Now, this particular enzyme, pyruvate dehydrogenase, it requires the presence of magnesium ions as well as TPP as a cofactor. It is thiamine pyrophosphate. Now, the next step is where the acetyl TPP complex, it undergoes oxidation, right? The acetyl TPP complex, it is a like undergoes oxidation where a 2-carbon acetyl group gets transferred from acetyl TPP to lipoic acid. Okay, so this particular reaction, it needs the presence of lipoic acid, or what is known as a lipoate, right? So it is this particular what you call product, that is acetyl TPP, it gets converted to S-acetyl lipoic acid complex, and the lipoic acid which is required for the particular reaction to be catalyzed, it remains bound to the dihydrolipoyl acid transferase, okay, or transacetylase, dihydrolipoyl transacetylase. So this is what happens during the process. Fine. Now the next one, it is actually the transfer of acetyl group onto the coenzyme A. So transfer to coenzyme A, resulting in the formation of acetyl coenzyme. Okay, so here what happens is the acetyl lipoic acid, which is produced after the oxidation process. From there, the acetyl group is getting transferred to the coenzyme A, resulting in the formation of acetyl coenzyme A and product, it is a dihydrolipoic acid. So acetyl coenzyme A and dihydrolipoic acid are produced as a result of this particular reaction. Okay, the what you call the enzyme involved is the acetyl transferase over here. Okay, now the dihydrolipoic acid has to be converted back to lipoic acid so that this reaction continues, right? Because here the lipoic acid is required for the process, isn't it? So the dihydrolipoic acid has to be converted back into lipoic acid, and this conversion, it is complete. This conversion is made possible by the enzyme dihydrolipoyl dehydrogenase, and this requires the coenzyme FAD. So lipoic acid and FADH2 are produced as a result. So dihydrolipoic acid, it gets converted to lipoic acid, and during this process FAD, it gets reduced. Okay, so this is the whole process taking place. So in a nutshell, it is like pyruvate, it is getting converted to acetyl coenzyme and carbon dioxide is produced during the process. And for the whole process to take place, what is required? Pyruvate dehydrogenase complex, the enzyme is required, which is formed of 3 different enzymes: pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. Okay, here you can see what all are the cofactors required. It is coenzyme A. For the last process, that is the transfer to the coenzyme A, they require coenzyme A. Okay, then they require NAD and FAD, where NAD is getting reduced, and then lipoic acid. I hope you remember in the second step during the oxidation, the lipoic acid is required, right? And for the first step, they need the presence of magnesium ions and thiamine pyrophosphate. So this is a whole process. Pyruvate, it is getting oxidative decarboxylation. It undergoes oxidative decarboxylation and oxidation. Okay, to result in the formation of acetyl coenzyme. And the whole process, it takes place inside the mitochondria. Now this is the enzyme here. Okay, the pyruvate dehydrogenase enzyme complex here, which is composed of 3 enzymes. The E1 is responsible for decarboxylation of pyruvate. You can see this is the pyruvate and carbon dioxide is released. The product produced as a result, it will take up oxidation. During the oxidation process, the NAD is getting reduced and the product, it becomes oxidized. Okay, this oxidized product, it will be taken for the next step, where the acetyl group of the product, it is getting transferred onto coenzyme A, and we get the acetyl coenzyme A and the dihydrolipoic acid as a byproduct. This dihydrolipoic acid will be converted back into lipoic acid so that the reaction can be continued. So this is what happens here. Okay, the what you call the pyruvate plus the NAD plus the coenzyme A, it will give rise to the acetyl coenzyme A, NADH and H plus, and carbon dioxide. So this is a whole step. Okay, so we have the pyruvate getting converted to acetyl coenzyme A, and it requires pyruvate dehydrogenase complex, which is a enzyme complex formed of 3 separate enzymes, and each of these enzymes are responsible for decarboxylation, oxidation, and the transfer of acetyl group to coenzyme A and resynthesis of the lipoic acid. So all these 4 are the steps involved in pyruvate to acetyl coenzyme A conversion. Okay, fine then. Thank you.